Hello everyone and welcome to today's webinar, Influenza Vaccine Viral Protein Profiling for Viral Potency. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SCIEX. To learn more, visit them at sciex.com. So we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you are having trouble seeing or hearing this presentation. I'd now like to welcome our speakers. Dr. Steve Locke, BioPharma European Marketing and Market Development Manager at SCIEX and Scientific Advisor at Horváth Lab at University of Pannonia. And Dr. Patrick Buckley, a Senior Validation Specialist at SCIEX in the UK. Welcome, gentlemen, to both of you. Dr. Locke, you may now begin your presentation.
Hello everyone, and thank you for that kind introduction. Today we'll be talking about a method to profile viral proteins in influenza vaccines for viral potency. So I'd like to start the talk today with just going through the outline of the presentation. This application will use a method called CE-SDS, so I'll discuss what is CE-SDS. I'll then look at how it's being applied to viral protein profiling. And then we'll switch speakers and I'll hand you over to Patrick, and he will talk about the use of this method in influenza vaccine viral protein and the reason why they do this type of assay. And then we'll finish off with a summary. At the end of the presentation, we'll be open up for questions from the audience. So what is capillary electrophoresis? Well, a capillary electrophoresis is a separation of analytes, proteins, small molecules based on their charge. And it usually happens in a capillary. The capillary widths are typically 25 microns to 100 microns internal diameter. It's an analytical technique developed originally by Beckman Coulter, which has been around now for well over 20 years. And it has quite a diverse application range. It's complementary and an alternative to liquid chromatography, and it really is well known for providing high resolution separation, and it acts as well as an automated version of slab gel electrophoresis. And in this slideshow, you'll just see a cartoon version of really how it works. So basically what you do with CE or capillary electrophoresis is that you apply a voltage across the capillary, and then you have a detector which detects your peaks. And this is shown in the next slide, and again, this cartoon. So basically, the capillary in CE sits in two buffer reservoirs of electrolytes. So you fill the capillary with electrolyte solution. And then you have a cathode and anode at either end of the capillaries, and then you apply a voltage, and what happens is the peaks migrate through the capillary, and they pass through a detector window. And those detectors could be UV, PDA, or laser-induced fluorescence detection. Now, in this particular application, we're looking at a technique called CE-SDS, which is capillary electrophoresis SDS. And basically, this is used throughout the biopharma industry, actually, for profiling proteins. Proteins by their size. And how this works is that you take the protein sample, you denature it. So basically what you're doing is breaking up the disulfide bonds, the hydrogen bonds, and the val-to-val interaction. So you sort of break up this secondary tertiary structure. And then you add SDS, which is sodium dodecasulfate, to the protein, and this coats the protein, adding a negative charge to the surface, okay? This helps also the denaturation. And so basically all the proteins present in the sample are then coated with SDS. And then what you do is you separate these coated proteins down a capillary full with gel. So it really is an automated version of gel electrophoresis. And as I said, this application has been used in the biopharma industry for years now for profiling monoclonal antibodies and this type of biotherapeutic medicine which is produced by the biopharma industry. And again, this slide shows you a cartoon version of how you do this. With the, if you look at the protein being unwound, and then the addition of SDS, which coats the surface of the protein, and then we separate these coated proteins by passing them down a gel sieving matrix, which is held in the capillary under a voltage. This technique is highly resolving, and basically what it does is separates the proteins based on their size. So it's a size mapping. The gel aids the separation, and it sort of sieves out. And what you'll find is the larger proteins move slower down the capillary, and the smaller proteins move faster down the capillary. Okay. And if I move on to the next slide, you'll see some examples of this and how it's been applied to the biopharma industry. So here you see typically a protein molecular weight size standard, which has been run through CE-SDS analysis. And here you can see that the peaks come out in the order of the size. So as the size gets bigger, the peaks elute or migrate later through towards the detector and detected later. And this can be plotted, so you can actually calculate the molecular weights based on the CE-SDS analysis of the proteins. And on this slide, we show the application to two biotherapeutic compounds. One is EPO, erythropoietin, and this is highly glycosylated. But again, you can see in the top example, the separation of the EPO, and this is, it's actually a method used throughout the biopharmaceutical industry to profile size variants of EPO. And you can see the comparison of that with a gel electrophoresis, slab gel electrophoresis plate next to it. One of the benefits of doing this type of separation based on charges at CE is very easily automated, is highly reproducible, and gives you more accurate peak areas than slab gel electrophoresis. And this has also been applied to the analysis of monoclonal antibodies like IgGs, and this is shown in the bottom example. And here again, you'll see a gel electrophoresis plate where the color bands mark the migration of proteins through the gel. The darker the color, the more concentrated, the more protein present in that band. And you can see the counter there next to it on the right-hand side, the capillary electrophoresis trace in the CE-SDS analysis of the same sample. And you can see, obviously, the areas are more easy to calculate from the automatic integration you get with CE. And again, this method is highly reproducible. So basically, CE-SDS has been used in the biopharma industry for well over 10 years, and there's ring trials to show how reproducible this method is in monoclonal antibody analysis. The next step, the next section I want to discuss is how this has been applied to viral protein profiling, and this is something which has happened only over the last couple of years as the biopharma industry moves to more complex next generation biologics, including things like gene therapy, biopharmaceuticals, which use viral vectors as a way to transfer genetic material, the active ingredient, into patients. And CE has actually been used to profile the proteins present in the viral capsids of gene therapy samples. So it's been used in profiling these viral proteins. And again, if you look at sort of this area of biopharmaceutical production, again, SDS page or gel electrophoresis has been used already for viral protein profiling. You see an example here on the far right-hand side where it separates the viral proteins of an adeno-associated viral particle by SDS page. You can also apply methods like reverse phase HPLC to separate viral proteins. However, the reverse phase HPLC method suffers from a lack of resolution, so you do have sometimes trouble in separating the peaks to give you correct integration of the peak areas. And SDS pages is really, again, it's very difficult to get an accurate value for the peak areas and the ratio of the viral proteins present in the sample, so it's more semi-quantitative in nature. With CE, however, and capillary electrophoresis, again using CE-SDS, the viral proteins are baseline resolved separated. So it allows you to more accurately calculate the peak areas for the viral proteins and therefore calculate the ratio of the viral proteins present in the sample. And this is important for adeno-associated virus because these viral proteins, which are labeled VP1, VP2, and VP3, this ratio gives you an indication of the potency of the adeno-associated viral particles. So one of the other benefits of using CE in this respect as well, and this is moreover than HPLC, is that you'll find that with CE you actually replenish the separation media between every injection, and you clean the surfaces of the inside of the capillary between every injection, so there's no carryover. And again, this is really important when you're generating reproducible data. That the removal of carryover is essential. And I know from prior experience myself that the LCMS or LC analysis can sometimes suffer from carryover, and CE generally does not suffer from carryover at all, and it's easy to remove. And this is because the capillary is used as the injector as well as the separating column, and it doesn't have any connectors like you have in an LC analysis where you have an injection valve which has connectors, LC connections to the column, for example, where carryover can happen. And this approach is very linear for looking at viral proteins. And here you can see a linear trace. This is done with UV analysis. So here you see that it's linear from 5 times 10 to 11 gene copies per mil up to 10 to the 14 gene copies per mil. And this is standard for CE, so it produces a very linear trace. One of the problems sometimes with viral protein analysis is sensitivity, and this depends on the sample. So often you need to increase the sensitivity for CE analysis, so UV is not sufficient, in which case you move to laser-induced fluorescence detection. And in this next slide, we show the traditional way of doing this laser-induced fluorescence detection, and this is not only for influenza proteins, but also for AAV samples, which are a lot lower concentration. And here you have to prepare the proteins. So typical approaches for preparing the proteins, which is basically derivatizing the proteins so they can be detected by the lift detector, is to add derivatizing reagents to your samples. Typically, these reagents were examples like FQ or TAMRA, and this complicated the process to some extent because it added a variable and also involved you doing some sample prep, including reconstituting the sample in different buffers for the FQ to work. However, this really did improve the sensitivity, and so what it showed that the sensitivity was improved by 2 orders of magnitude, by nearly 100-fold, if not greater, when you moved across to laser-induced fluorescence. In this particular example, in this method used for viral proteins, when we did use this FQ derivatization, we actually built around a pre-built cartridge, so it's like a standard capillary, and again we use our standard SDS kit. So what you find when you move to laser-induced fluorescence, so when you tag the proteins, it doesn't affect the pattern you see. The patterns are still the same whether you do laser-induced fluorescence or whether you do UV detection. It just basically increases the sensitivity. But what happened last year was that we looked at this derivatization process for laser-induced fluorescence, and we devised at SCIEX new chemistry to simplify the derivatization of the proteins. And this chemistry was based around a commercial dye you can buy from Active Modif or Sigma Aldrich. And this dye is a chromium dye. It's a pyridinium-based dye, which is very easy to label the protein. So the sample process now is very simple. You would do your standard denaturing of the sample. So you would basically denature the sample to break up the tertiary structure. So that's heat denaturing with the presence of SDS. And then basically to label the protein, all you simply do is add the dye and heat for an additional time period. And then your sample's ready to analyze. Typically, at that point there, we simply dilute the sample and inject on the CE system. This really simplified the derivatization process and made LIF detection a lot easier to use. And on the right-hand side of the slide, you'll see an example of this, again applied not to influenza proteins in this case, but applied to adeno-associated proteins, adeno-associated viral proteins. And this is a serotype 8 sample. And here you can see that the viral proteins are detected at a concentration of around 10 to the 9, where previously the limits of detection were around 10 to the 11. So again, as I mentioned earlier, this gives you a 100-fold improvement in sensitivity. But the profile map, the image you see, the relative ratio haven't changed. It didn't impact the ratio you were seeing. So this is a really good way to improve the sensitivity of the assay. So that gives you an overview of how this CE-SDS approach has been applied to protein profiling and also to viral protein profiling. But this presentation is about how it's been used particularly for influenza viral protein profiles. And I'd like to now pass you over to Patrick, who has developed this method in collaboration with SCIEX at SCIEX to actually use this approach in viral protein profiling. So over to you, Pat.
Hi, Steve. Thank you for that. It's a great introduction. So, yes, as Steve said, the second section of today's webinar will cover the development of CE-SDS for the analysis of influenza viral proteins. And this is to replace SDS page. And this development work has been carried out in collaboration between myself at SCIEX Liverpool and Stephen and his other technical colleagues at SCIEX. And I'd like to say that SCIEX has been instrumental in progressing the CE-SDS assay in Liverpool. So as an introduction, influenza viral proteins consists of a number of different proteins, and particularly they can be characterized by their glycoproteins. And these glycoproteins include hemagglutinin, HA, and neuraminidase. And these hemagglutinin and neuraminidase proteins change depending on the strain that is present in the virus and in the vaccine. So hemagglutinin is a class 1 fusion protein which has multiple functions and is important in binding the influenza particle to the target cells. And previously, a long time ago, the SRD test was developed in the 1970s, and this uses the quantity of the HA proteins which are present in a sample to provide an indication of potency. Now, the SRD test is subtype and type specific and is immunological reaction based, and sometimes has challenges with cross-reactivity, as can happen with immune response based tests. So along with Stephen and a couple of the colleagues in Germany and Poland, we introduced this method and developed it. So we used a fluorescent chromio dye, which Stephen talked about earlier, P503, and we actually used a reverse polarity method in the CE system, which meant that we could shorten the sample analysis time to 12 minutes. And this shortened time allows us to test a larger number of samples more quickly, and it also exhibits suitable resolution of the major hemagglutinin peaks in our 4 seasonal influenza strains present in our vaccine. So you see here on this in this figure, parts A, B, C, and D are the different seasonal strain subtypes. And when we actually prepare the sample, we use decarboxylation and reduction, and this breaks down the hemagglutinin protein into 2 sections, which is hemagglutinin 1, hemagglutinin 2, which we call HA1, HA2. And these are well resolved in the SCIEX system for us. As part of this development work, we ran 12 replicates of each of these seasonal strains, and an example is here in this electrophoregram of 12 replicates superimposed on each other, and you can see there's excellent reproducibility with minimal shift of migration time. And if you look at this further, you can actually measure the RSD of migration time and of the corrected area of HA1 and HA2. As you can see, the migration time is really consistent, and the corrected area has a RSD of below 5% for HA1 and HA2. This again shows how consistent these 12 separate injections are on the system. So to summarize, the method developed, we used a 90 sample format, which separated and identified HA1 and HA2 fragments in an automated fashion. The lift detection improves sensitivity and allows reduction of sample loading, which in turn reduces the capillary cleaning needed between injections, which speeds up the analysis. Separations are run in 12 minutes, and we've shown that the CE-SDS assay and analysis developed is robust and reproducible for influenza viral protein profiling and can be applicable to different viral strains.
Great, thank you, Patrick, for sharing your data today, because I think it's really important to share this information with the greater community. And thank you as well for working with SCIEX to develop this assay within SCIEX in Liverpool. I also want to acknowledge the team I work with within SCIEX, including Marcus, who's based in Germany, Marcin, who's based in Poland, and Sam, who's based in the UK. I also would like to thank everyone for attending the webinar today, and hopefully you've got an insight into how CE-SDS with lift detection is now being used in areas other than antibody production within the biopharma industry. And at the end of this presentation, I would like to open up both Patrick and myself for questions, and I also would like to thank LabRoots for hosting this webinar today.
And I want to thank you, Steve and Patrick, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. It looks like we already have some great questions coming in from our audience members. Our first question is, can this approach replace DLS for size and ZP?
Okay. Do you know much about this, Steve? Or, I mean, I've got a bit of experience about.
Yeah, I think you know a lot more about this than I do, Pat. So over to you.
Okay, that's fine. So I would say no, because it really looks at sort of proteins in general rather than large organisms or fragments. So I think it'd probably be an excellent sort of method to use alongside DLS to give you more information about your product or whatever you're working on.
Wonderful, thank you. And what is the minimum MW for this approach, and can it be used for 15 to 30 mer peptides?
Shall I answer that one, Pat?
Yeah, that's fine, yeah. Okay.
Yeah, we typically, I think you can use it 50 to 30 mer peptides. There is some residual dye which comes out, and it comes out around about molecular weight of a couple of thousand Daltons. So as long as your peptide is bigger than that, and 15 to 30 ml it should be fine to use a similar sort of approach with large peptides.
Yeah, I think the system I've been using of your SDS molecular weight kit comes with standards which start at 10 kilodaltons. So again, I guess it depends what you want to do with your samples. If you want to see them on the system, then it should work. But if you want to get an accurate molecular weight, then I would say they're not large enough.
Yeah, because I think with the 15 to 30 ml should be, if it's 30 ml, definitely be 10K and above, so it should be fine then.
Yeah.
I know that the residual dye part is a lot less than the 10 kilodalton marker.
Yeah,
you have
absolutely.
some room there as well.
Thank you, gentlemen. And now, can CE-SDS be used as a purity assay? And if yes, how robust and sensitive should it be when compared to SDS page?
It actually is. So I'll answer this one, Pat. Just a quick question. Yeah, it is being used as a purity assay for size mapping already for monoclonal antibodies. And sensitivity is a good question because I suppose it depends on protein loading. On an SDS page, you tend to load a reasonable amount of protein for like a 2G gel separation. And with CE, you load a lot less. You're typically injecting nanoliters a sample. If you look at this sort of approach, within SCIEX, I'm working with Marcus Heinl to look at sensitivity limits. We're down really analyzing standards which are around about a nanogram per mil level. And when you're only injecting nanoliters a sample, it really is more sensitive than a standard, I would say, SDS page approach. But it really depends on loading, because you say you load more on SDS page.
And you can see also on the slide 22, you can see how reliable the data is if you do multiple injections as well. So that give you an idea of how robust the assay could be.
Yeah, thanks, Pat.
Okay.
Thank you both. And I also want to thank our audience for these great questions coming in. Our next question from an audience member: how is the separation efficiency, the resolution between VPS compared to SDS page? And also, can you comment on the sensitivity comparison of these two techniques?
Ooh, Pat, do you want to answer or shall I?
Probably best for you, Steve, really, because I've not had that much experience of this yet.
Yeah.
Regarding to our influenza proteins, the separation efficiency is absolutely fine. It's good resolution, but again, it probably depends on your analyte, really. And regarding sensitivity, I guess, Steve, have you got more ideas about sensitivity?
Again, sensitivity, I know this approach can detect, say, solutions which you have less than a microgram per million of protein. So it's a pretty sensitive approach. I know we do it for example, if we're doing impurity profile in an antibody, which you can also do by SDS page, we inject just a couple of, the concentration of the sample is microgram per mil, and then we're injecting like 1% impurity. So that gives you an idea of the sensitivity. I think the resolution is also, you got to remember, when you're staining SDS page gels, the resolution also depends on the staining, right? And here you're basically doing left detection, so it's far more reproducible and calculating the area, and you can integrate your peaks a lot more accurately with a CE separation compared to a standard SDS page approach. So I think probably the resolution is better, to be honest, simply because it's a lot easier to integrate the peaks when you're doing CE rather than doing an SDS page gel.
Yeah.
Thank you, gentlemen. Now, is it cost effective than SDS page and RP HPLC, or is the protocol easier than the SDS page?
I think from my work over the last few months, I think look at the second part of the question. The protocol is probably similar to SDS page for sample prep. It's really very similar. You don't need any extra reagents. It might possibly be a bit quicker than SDS page preparation. For my work with influenza, I was having to decarboxylate and reduce the samples first, so that adds an extra step to that, but then I would do that anyway for SDS page. So that would be the same. Cost effectiveness, I would suggest that it's more cost effective than SDS page, and possibly more than reverse phase HPLC as well. I know in our lab we had problems with columns blocking in HPLC, whereas I've not had any issues with capillaries misbehaving in CE at the moment. So I think we certainly suggest that it's on a par with HPLC and more cost effective than SDS page. Would you agree, Steve?
I think that's right. I think what you're right with the column blocking. I think with CE we sort of use an open tube which we fill with gel, and then we re-clean the tube between every injection, right? So it's not filled with particles like you have with the HPLC column, so there's less chance to block. So capillaries tend to be pretty robust, and you can run it for loads of injections. And you've got to think of the volume of solvents used with HPLC. Your solvent usage is significantly higher than your background electrolyte uses for CE. You typically, with buffers, right, Pat, you only fill the buffer tracer with a couple of mils of
absolutely.
solvent.
You use that. Yeah.
Thank you so much. And we have time for a few more questions. Is there a specific ratio for concentration of the dye to the concentration of target protein?
We haven't really tested that, have we? I'm just trying to think. We always use the dye in excess, and we tend to use the same protocol every time. We've used it for several different applications now. I know you can dial it down and decrease the concentration, but typically it's only 50 to 100 times the protein concentration, I would have thought, off the top of my head. Does that make sense?
Yeah, I think so. Yeah, it's definitely, we use an excess of it because there's always a peak that comes off first of the unused dye. So yeah.
Thank you very much. Now, how many different proteins make up influenza virions? And would CE-SDS work for viruses like lentivirus that are made up of 10 to 15 different proteins?
Okay. So for influenza, for our system, when we manufacture the vaccine, there's not that many proteins that make up our product. So I think we lose quite a lot of the proteins in the process. So when we test like our product is like is a non-inactivated split virus. So when I look at whole virus, there's only 3 or 4 major proteins that are present anyway. And then when we looked at the final purified vaccine, there's mainly sort of 2 proteins that make up the vaccine. So with regards to lentivirus, if it made up to 10 to 15, again, it would depend on really like a weight of the different proteins. If they're all similar sizes, then you might struggle. But if there's a broad range of sizes, then you'd probably be okay. Would you agree, Steve?
Yeah, I can actually answer the lenti question. We're actually looking at lenti within SCIEX to run lentiviruses with a similar approach, and we actually can separate out the proteins present in the viral capsids for lentivirus. We should be releasing some application notes on that in the next couple of months. So if you're really interested, just watch the website because I know we're releasing some data out on that in the next couple of months.
Wonderful. Thank you so much. We have a few more questions that are coming in, and I just want to thank our live audience for these great questions. Gentlemen, can this method that you use be applied to other viral particles?
Shall I answer that one, Pat?
Yeah.
Yeah, it definitely can. I think it depends, again, on the complexity of the viral envelope. I think early in the presentation, I discussed adeno-associated virus, which is a gene therapy viral vector. We've actually used it for a lot of different types of serotypes of AAV. It's been used in a couple of sites now within the UK to profile AAV serotypes, again, for a similar sort of application for viral potency. As I said to answer an earlier question, we've also used it within the research labs to look at lentivirus as well. And so we're releasing data on that in the next month or two. So far we've looked at, say, influenza, lenti, AAV. We haven't yet applied to adenovirus, but I presume it will work for adenovirus as well. I suppose the key thing here really is that you have to break up the capsid. So what we found with AAV is that you have to use different temperatures to reduce the capsid in order to pick up the proteins. So some serotypes require a higher temperature to reduce them, so you can get hold of the protein to do the labeling and detection. But that's the only thing we've encountered so far when we've applied this to different viral particles.
Thank you very much. And can CE be used for other areas of vaccine quality control?
I would suggest, yeah, I would think so. I don't see why not. It's something we're at SCIEX looking into. Because we have 2 major products. One's made in cells, one's made in eggs. So in our products, we of course have egg proteins and cell proteins that we don't necessarily want. So we're certainly going to start looking into those areas as well to see if we can replace any ELISAs, for example, for egg proteins. We could use CE instead. So yeah, I would think it should be.
Yeah, and there's a similar application which I'm working with another customer where we're looking at with a conjugate vaccine, where you conjugate polysaccharides to a protein, and the proteins can be things like toxides, right? So you need to, with that typical type of vaccine, you need to be careful how much of the free protein is present. It's similar to what Pat was saying about egg proteins being present, and we use a similar approach to look for trace levels of that protein present in the product, and that's a sort of application that Pat was discussing as well, is to look for trace level protein contaminants present in the viral vaccine or the vaccine product at the end. I think that's one application which uses honestly the same method, just adjusted slightly for the sensitivity. I think the other thing we can do is look at gene therapy applications. We developed a few last year in SCIEX, where we were looking at empty versus full viral particles, again in gene therapy. Maybe that's an approach to apply to other viral vaccines. Is to look at, I was thinking specifically at, we hear the news about viral vaccines used for COVID, right? And whether we can apply CE to look at whether those particles are empty or full with DNA. And that may be something we'll look into. So we're already using that sort of approach to look at gene therapy, AAV, say products, where we're looking to see whether those particles are empty or full of genetic material. So that may be an area where we could apply to other vaccines and that move outside of gene therapy. So that's an idea that we're looking into as well.
Gentlemen, thank you so much. We have time for one more question. This audience member says, thank you for this nice presentation. Is there a reference describing the details of the experiments presented today?
Yeah, I can answer that one, Pat. We are planning to release a technical note on this as well, which will go into more detail around the applications that Pat went through today. And I think that's still in our legal review process, isn't it, Pat?
Yeah.
Hopefully that will be out in the next couple of weeks.
Yeah.
Thank you so much. And would you gentlemen like to provide any closing remarks before we go? I'll start with you, Steve.
No, I think first of all, thanks for everyone for attending today. I saw the audience figures and it was really good, and thanks for all the questions. It's really helps when the audience engage with us and ask the questions. Pat and I are really open to answering any questions that the audience has. So thank you again. And for me, it's a thank you for Pat for allowing SCIEX to work with laboratories to present what Pat and I have done together at SCIEX.
Wonderful. And Patrick, did you want to add anything?
No, again, just like Steve, so thank you everyone for attending the webinar and for the great questions. And also, I'd like to thank Steve and SCIEX, as they've been instrumental in developing this assay for us over the last probably 18 months now. And so I think just like to say how good the scientists are at SCIEX and helpful. So if anybody's got any questions for SCIEX and want to get involved, I think it's a really good company to help them with their research.
Well, thank you both again, Steve and Patrick, for your time today and for your important research. And I'd also like to thank LabRoots and our sponsor, SCIEX, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everybody. Bye-bye.